Hi everyone, my name is Iman Hajirasouliha. I'm a faculty at Weill Cornell Medicine. It's a medical school of Cornell University. I'm also the co-director of our tri-institutional computational biology and medicine program. So if you're looking for a PhD study, you can apply to our program. So it's my great honor to be here and speak today. Thanks for sticking the last day. I always say this is my favorite meeting of the year, so I'm happy to be back. So today I'm going to talk about some actually new work in the lab on metagenomics analysis systems. But just before that, I want to sort of like go over some of the other projects in the lab. So just a very brief introduction to my computational biology lab. So we do a lot of different studies. So it's like a very diverse set of projects in the lab. So for the most part, my lab is focused on computational genomics and metagenomics. We develop methods for assembly, classification, human genome characterization, with also application to cancer genomics. But also more recently, we study a lot of AI applications in biomedical domains. Specifically, we are focused on women's health. So I work a lot on fertility issues, ovarian diseases these days. We also work on digital pathology, classification, and also, again, human genomics with AI. So today, I'm going to mostly focus on metagenomics. And specifically, this is not sort of like a new algorithms sort of like development. It's more like a pipeline, which is, we call it CAMP. And the idea is, okay, so I'm going to go give a very quick introduction, but I don't think I need to because this week we heard a lot about metagenomics. We are all agreed that metagenomics has become an important field in bioinformatics, and specifically, microbiome communities typically are very complex. There can be dozens to thousands of species. Some of them known, some of them unknown. So it's really important to characterize these species. And also, we all learned this week that we are generating a lot of data, like a lot of data, right? So we really need precise and reliable workflows to identify these species, study functional roles. So it's important to have these robust workflows. So this slide is like a very busy slide. I don't want you to read all of it, but I have put 19 recent pipelines. This was, I guess this slide was updated this April, but I'm sure there are more since April. So you can see, there are tons of workflows and pipelines for analyzing metagenomics. For example, they are, and for good reason, I guess. Like there are all useful pipelines. People use them, and in fact, a lot of labs they have individual in-house pipelines. So when I talk to some colleagues who are doing metagenomics, they actually have their own pipelines in-house, so they don't even use any of these existing tools. So for example, there are pipelines for metagenome assembly, binning, pipeline for classifications, several pipelines for each of these tasks. So the thing is, some of these pipelines became sort of popular, but I guess most of them are not very popular. And the idea is, okay, so metagenome analysis is sort of complex. So you usually start from raw reads, but there are tens of tools that you need to use to get, like to your downstream tasks. For example, if you want to do a metagenome assembly or you want to get to MAGs, you need to utilize, several tools in your pipeline. But obviously, these tools, all of them, come from, they have like their own dependencies. They might have conflicts. People update tools, so they may, likely phase out in a matter of years, usually. And in fact, a lot of existing pipelines are these generally all in one pipeline. So people want to be helpful, so they want to make things easy, especially for, I guess, non-computational biologists. So they want to, provide a pipeline, just a one-click pipeline. So you start with your raw reads, and then you get to your downstream analysis. But this is a bit challenging, especially for these complex pipelines, because you need to do version controlling. It's hard to maintain it. So if developers leave, so you need to, there are newer versions of each pipeline. Essentially, if you are developing newer things, it's hard to, add to the existing pipeline. So when I was a grad student, we were sort of told that, as I said, it's good to have these one-click pipelines. But these one-click pipelines don't give us a lot of flexibility. So for example, if you want to start from raw reads, do quality control, assembly, taxonomic classification, functional characterization, mag binning, you can have all of them in one single-click pipeline, but this would, substantially limit your flexibility. So there is not a lot of additional development potential. There are many chances that these things fail when you have this big one-click pipelines. And you don't have a lot of flexibility with parameters. So an alternative approach is what we call a modular approach. So we have several stop points, usually after each task, and then we call them a module. So we have for each task, for example, if it's a quality control task, we have a module, and then we stop after this module, and we can essentially design consistently across all these modules and document each module separately. And for visualization, we can also have a built-in visualization for each of these modules. So this would help, especially non-computational biologists, look at the data right after completion of each task in a harmonized visualization with a visualization tool. So there are some additional benefits if we sort of switch from these one-click pipelines to modular pipelines. Sometimes you don't want to run the whole pipeline. You just want to choose a workflow for a specific analysis. So you may just download a couple of these modules, in a chain and then run them. As I said, there are these built-in soft pauses, so you can explore the results after each module. You can essentially look at these modules as benchmarking tools. So if you are developing new modules, new tools, you can, do benchmarking. And this also gives you a flexibility for future developments. So you can develop new modules, even for completely new tasks, and add this to the whole environment. So we developed this core analysis modular pipeline. It's called CAMP. And the goal of the project was, as I said, to make metagenomics robust, reliable, flexible, and reproducible. So this is mainly work of my graduate student, Lauren Mack. This is a picture of us in CGSI 2022. She's actually graduating very soon, so she'll be on the job market in a couple of months. So the idea was to develop these modular templates. And we also did some pilot analysis on meta-sub sequencing data to show the utility of these modular environments. So Metasub is an international project led by a colleague of mine, Chris Mason. It's a very ambitious project, mostly looking at subway and transit system across the globe. So there are more than 100 cities involved in 7 continents. So you would want to do molecular profiling of samples across all these transit systems. So the tool we developed CAMP is already available on bioRxiv. The paper is under review. You can check it out. And we have all the modules available on GitHub, meta-camp. So here is, again, a busy slide, but I'm going to show you, all the modules that we developed in CAMP. So you can see all these different boxes, different tasks, individual modules. So some of them, mostly on the left, they are colored with light red. These are modules that normally you would use at the beginning of your pipeline. For example, you can see short read quality control if you have your input is short read, or it can be long read as your input. So there is long read quality control. Then some of the boxes here, they have more like medium red color. And these are the intermediate modules. For example, you can start with short read quality control and then go to short read assembly. So you get assembled context. Or you can do hybrid assembly, both short read and long read. Or you can do mag winning, for example. And there are some of them with dark red, and these are more terminal modules. For example, can be an end result of a taxonomy assignment, or it can be gene cataloging. You can assess the quality of your mags, and so on. So, and then you can also see on these figures, some of these modules, some of these boxes have these bar plots, and that means they have this built-in visualization tool. So you can actually, once you run the module, you can automatically see the results in bar plots, or if one module is running, for example, several different taxonomy assignment tools, for each of them you can actually see the results. And the ones that have the cylinder, that means they actually download data. So it may be like a database you need to download for your taxonomy profiling, or some additional databases that you need to download. So that's the general framework for this CAMP. Again, as I said, this is a plugin whenever, pipe out whenever tool. So you can essentially pick the module that you need to, right? So you don't have to do everything at the same time. So for example, you can essentially walk on different passes of these complex network now, right? So you can, for example, there's an application, you have short read and long read, so you can do, for example, a hybrid assembly or short read assembly, and then do the mag binning. Or later, you can do additional modules on different chains of this your workflows. Okay, so just to summarize this, CAMP addresses some of the challenges we have in metagenomics analysis. So one challenge was, as I said, we have complex data sets. The pipelines are huge. There are like lots of tools that you need to do. And then CAMP gives you flexibility to, for example, do a lot of different analysis. For example, in this case, more than 9 different analyses in a self-contained, harmonized modules. So there are problems such as resolving species-level genomes. It's still very challenging in metagenomics. But with CAMP, you can tackle this problem, especially if there are easy-to-use, updated tools. You can essentially embed this into the pipeline. Instead of doing manual curation for a lot of the downstream analysis, CAMP can do this automatically. You can also take advantage of this built-in data viz. So you can essentially look at input and output for each of the modules. And finally, it's actually not that hard to run this. Okay, we don't have like one-click pipeline, But you only need 5 commands to set it up, and again 1 command to test or run it. So just 5 lines. So you can, and essentially there is a documentation, so you can actually easily run it. Even there are for users who are not expert or they are not computational biologists. Again, this is one of the modules. For example, the short-read quality control. There are several steps embedded in this module. For example, you want to do filtering of low-quality reads. We want to remove host reads, correct sequencing errors, and the thing is like these are all very standard across all modules. So they look all the same, and there are all 5 commands to set up, and then one command to run. So this table compares CAMP with several available metagenome analysis pipeline, namely Atlas, MAG, Snake workflow, Muffin, NF Core MAG, and NBIS Meta. So I'm going to slowly go over this table. So CAMP is done with SnakeMake, just because we were mostly familiar with Python. So we did it in the SnakeMake. Muffin and NF Core does it in Xflow. I guess we can also have an Xflow version of CAMP. The highlight is CAMP is modular. The other pipelines are one-click pipelines. We have done install testing for all the modules in CAMP. There is documentation. It's very extensive. Some of the other tools, they also have like extensive documentation, for example, NF Core. But CAMP also has a lot of very extensive documentation for all the modules that were implemented. So you can check out the GitHub. We do have data visualization, as I mentioned. And for some of these tasks I listed here, for example, long read QC, hybrid assembly, we already implemented modules. We still need to do co-assembly and co-binning, so that is under process. So MAG binning, as you can see, these are, so we are doing 6 different tools for MAG binning, plus 1 tool that is sort of like an aggregator, more than any other existing pipeline. We also do short-read taxonomic classification and virus phage detection. And again, you can add any additional module that you want. So I'm going to go over quick examples. I think mostly I covered them, but just want to highlight a few examples. For example, if there's a user, they have a short-read data set, and they want to do some analysis, for example, do taxonomic classification, they can only download two modules, right? One to do the quality control, QC, and then one to do the short-read taxonomic classification. And then this tool would give you a result in a nice visualization manner. And then another user may develop a new short-read taxonomic classification tool. So you can add that module to the environment. You can actually use it as to benchmark with other existing tools in that module, and then you can see the merge report. And then if someone, for example, wants to do MAG reconstruction, they can only download the binning and the MAG quality control modules, and then add this when you have the result of the short-read assembly. So I'm going to give some results obtained by CAMP on a pilot study we did with the Metasub project. But we are, essentially like people in Metasub are now using CAMP for the most part. So this is 10 randomly selected samples These are from subway stations across New York City, mainly Manhattan, Queens, and Brooklyn. So you can see, some information on the samples, sampling time, the train lines, the name of the station, and the method for swabbing. So if they did the seats of the train, or they did the handrails, the number of reads. So you can see it's actually very variable. So some of the samples have a lot more reads compared to others, and the total size of the base pairs. So you can see, for example, I highlighted a couple of samples here. Sample number 3 had the least number of reads, only 3,600,000, but sample number 7 had a lot more, 46 million reads. So we can use CAMP, do the shorted quality control, and then shorted assembly. So here is some results of the QC, and these figures are generated automatically by CAMP. So you can see I'm highlighting sample number 3 and sample number 7, the one with the least amount of data and the one with the highest amount of data across these 10 samples. So you can see, for the most part, we are retaining most of the reads, 87% of the reads, or 81% of the reads, 80% of the reads. And most of the time, as you can see here, and then this the graph shows you, we lose reads at a low quality removal step. So this was generated by CAMP. We can also look at de novo assembly, because we also use that particular module. And for that, we start with the shorted quality control, the output of the shorted quality control. So you see the graph for all the samples on the left. And you can see metrics of different assemblies for each of the samples. For example, the number of contigs, the number of bases, and the mean contig sizes. So you can see, for example, for these samples, as you, I guess, mostly expect, the samples with the highest number of reads would result to better assemblies. I guess one exception here is sample number 4, which did not have the most number of reads, but the number of contigs and the contig sizes, the number of bases covered is the highest. So there might be some explanation that maybe there are not a lot of duplicate reads, or like more unique read in that sample. But again, you can see, CAMP would generate all these graphs for these assemblies. We can also do mag binning with the pipeline. So again, I'm focusing on these two samples, sample number 3 and sample number 4. And essentially, you can see for different parameters of minimum contig size, when you want to retain them for binning, the results are different across these samples. And for the most part, the number of reads by itself is actually not predictive of the assembly size. And obviously, there are a few of these contigs that would escape to the bacterial chromosomal lengths, right? So if you, for example, set sort of like a standard mean contig size of 1,000 base pair, you would essentially retain up to 10%, 8.8% of the contigs for binning. So this was the module that was used in CAMP. So the CAMP uses, if you want to do mag binning, we use a lot of different tools, as I mentioned. So 6 tools and 1 DAS tool, which is an aggregator. So you can see the results across different tools are different. So some of them, have higher number of bins. Some of them have less number of bins, especially, the DAS tool, which was a more conservative aggregator. But DAS tool generate, longer longer contigs in each bin. So you can do MAG quality control. You can look at, like a wide range of metrics, for example, completeness, amount of contamination, the clade separation score across, like different tools for MAG binning. So you can see, for example, in this example, on our 10 sample, we had 62.8% of our MAGs high quality based on the MIG-MAG standard. We can also do taxonomy classification. Again, another example of the modules in MAG, in CAMP. And you can see this is also an interesting result on this pilot study, because you can see that actually the choice of the shorted classifier is more predictive of a sample taxonomy composition. And for the most part, across all the ranks, species, genus, family, like all of them, you can see a lot of the tag size assigned uniquely, so for only one of the tools. Okay, so some more takeaway on this pipeline. I think I mentioned these, that a lot of times like these tools don't agree with each other, but you can actually see this as a result of CAMP in the visualization. There are like different binning strategies. They may give a different number of MAGs. We have this built-in database, and CAMP can coordinate among like all these different tools. Like 20+ tools is already implemented, so you can balance convenience and utility with CAMP. So what CAMP doesn't do at the moment? So we don't have the containerization in CAMP, so that's something that we may implement soon. We still don't have the co-assembly or co-binning. If you have want to assemble multiple species at the same time. There are still some analysis endpoint that can be added. For example, if you want to do metatranscriptomics or variant calling or strain inference. So there are some alternatives. For example, Muffin can do this. And finally, you can. Okay, this is still like easy to use, but we can use AI and LLMs to assist a user when they are dealing with CAMP or want to use CAMP. So this is an ongoing project in the lab. My postdoc sort of working on this. Wei, he should just join the lab. So the idea is so we call it boot camp. So this is an LLM assistant for CAMP. So basically, we still want to be very user-friendly. So even if it's only like five commands, you can ask an LLM, like a specific LLM, okay, I have this analysis, what kind of modules I need to use. And Bootcamp would give you an answer based on a large language model. So this is one example. She actually just gave it to me yesterday. So you can see, okay, I want to see. So one prompt is tell me about CAMP. So you can focus on the README of CAMP and the paper, and you can see would list like what CAMP does, all the modules that are implemented in CAMP. And then if you want to have another comment, I don't know if you can read it well, but if you want to have another comment, for example, how I can do a long read QC, it gives you exactly what module that you need to use to do the QC. So it's basically more focused on like a specific database for CAMP with the source code and the documentation and README, and also use a pre-trained available large language model with some agentic framework. So this is sort of like a design. It's preliminary, but it still works very well already. But we want to expanded to more troubleshooting and some additional interpretation. So just to summarize this part, so CAMP is a modular ecosystem for metagenome analysis. It's customizable, for anyone's project need. It's also freely available on GitHub, so you can download and use it. And we are also developing this bootcamp, which is the AI assistant. Okay, so I think in the last few minutes, I want to sort of highlight another project in the lab, which we call Microbiome in a Bottle, or MIAB. So the sort of the motivation is from this Genome in a Bottle project. This is a very ambitious project led by NIST, the National Institute for Standard and Technology. And the goal was to build a high-quality benchmark set and reference for human genome. So I was involved in some analysis for structure variation benchmark sets when I was a postdoc. So this was like a more than 10-year project now. So it's still going on. So they are adding more samples. But the idea was, because we have power to do a lot of sequencing, especially with newer technologies such as long read or linked reads, synthetic long read, you can generate a lot of data. So for example, a lot of high coverage short-read data, high coverage long read data, and additional sequencing. So essentially, NIST partnered up with all the technology developer, and they use a lot of different data set to build these high quality benchmark sets for human genome. So when I started my lab, I was like, okay, why not a microbiome in a bottle project? So can we do the same for metagenome benchmarking? So it's like a pretty ambitious project. So we worked with Zymo Research. So we looked at human gut microbiome. So we had a few samples from human gut. So our eventual goal is to have 4 gold standard benchmark data sets for metagenomics. So if someone is developing new tools, they can use this curated, high-quality benchmark set for evaluation. So one of the samples is now very complete. So we work with a lot of technology developers, and we also did a lot of sequencing ourselves. So we did 2 DNA extraction, 2 different protocols for DNA extractions, 2 technical replicates, and 5 ultra deep sequencing on these samples. So mostly Illumina sequencing for short read and Oxford Nanopore for long read. But we also work with some technology developers to generate these synthetic long reads or link reads with TelSeq and Loop Genomics. And we also had some high-C data from Phase Genomics. So these are 5 different technologies. Actually, mostly we use CAMP now to analyze this data, but we also want to reconstruct, isolate genomes from this Zymo community. So we're going to release the data very soon. So that's sort of like an announcement. So in a couple of months, we'll have the first data set release. So the goal is to support method development, pipeline validation, and so on. So I'm going to show you some high-level overview of what we did with all this data. So the goal was to generate these high confidence, high quality mags using like all these different technologies. So essentially, we use, for example, Illumina data and TelSeq, which is also a short-read sequencing method, but with some additional, like linked data. So to generate, these medium quality and high quality mags, and then we had, a lot of long read and loop read sequencing. So we use this blender approach in high level to do hybrid assembly and essentially get to circularize the genomes. So one main observation of all this data was that this multi-technology approach would be actually needed if you want to generate like these circular genomes and high quality assembly. And this is sort of similar to what we observed with the genome in a bottle. So for genome in a bottle, mostly long read data was enough, but still like you needed high coverage long read. In addition to that, you needed some short read and high coverage short read and other data sets to generate these high quality references and benchmark sets. So I'm highlighting a few of the results. So this is still work in progress, but just like a few examples of what we observed. It's interesting because almost 72.5% of the discovered species in our community were found in two or less, or one technology / extraction replicate. So there are a lot of unique species for a technology or a replicate. This figure sort of shows the distribution of the prevalences of different species which were observed using for example, one of the technologies is long read sequencing of Oxford nanopore. And you can see, like the distributions kind of look the same, but the prevalences can be actually different across different technologies. So you can see on this side, these are, like abundances for short read and link reads compared to long read. And you can see, like it's not always consistent. So each technology is, like capturing different prevalences. This is another result which is, I think it's interesting because we still have a lot of reads. So we generated a lot of reads, And you can see still, we have a lot of reads that are not assigned to any of the candidate isolates. So even like we map them to the reference or we map them to the constructed mags. Still like a lot of unmapped reads, both for short read and link read, and also for long reads. So for long reads a bit less, maybe 6-7%, but for short reads a lot more. Okay, so just a few more takeaway for this part. So I'm summarizing some of the results that we had. So with these large short read or link read data sets, for example, TELSEQ, the more read we have, the higher quality mags we can generate. So in like overall, like the mag quality didn't differ much with short read, link read with different extractions. But still, a majority of the discovered species, like 70% of them, were found in less than half of the biological replicates. So we had 71 high quality species classified as mags, but like they account for only like 72% of the Illumina reads and only 93% of the long reads. So there are still a lot of reads which are not assigned. We also did hybrid assembly. So we used the AutoCycle. So this is sort of like an overview, sort of like a complex pipeline sort of Lauren came up with. So using AutoCycler, but with adding additional data sets of short read for polishing and all of that. So I'm not going to go through the detail for this pipeline. Just highlighting some of the results that we can see for some of the species that we actually can get to this circular DNA when we do the merging and also combining different data sets. So this is one table sort of showing, like with this hybrid approach, using the long reads, we can actually generate, like the higher quality mags, and also we can capture some of the, like challenging, rRNA genes. So it's so we can't capture any of those when we only did mags using short reads or link reads. Okay, so this is a few more takeaway for this part. So if we did the hybrid approach with all the technologies, we can actually get this rRNA material. It's important, like what kind of parameters we use for long read minimum sizes in the assembly. And like even with only long reads, and this is something that we observe and we had to deal with it, that like some of these long read assemblies are still fragmented or incomplete. So I think I'll stop here. I want to thank my lab, especially Lauren, who was leading all these efforts in the lab in the past couple of years, but also a lot of other lab members contributed. I want to highlight Chris Mason's lab, collaborator at Weill Cornell, and the Metasub Consortium. I also want to highlight some of these technology developers who work with us. Zymo Research processed the samples, but Phase Genomics, Loop Genomics, UTS, they did a lot of sequencing for us. And I also want to highlight NIGMS for funding. Thank you.